everyone. Welcome to Crossroads. I'm going to be your host today, Alyssa Adams, and I am at Cross Labs here in Kyoto, Japan. So basically, Crossroads is our monthly fun forum where we invite all sorts of speakers from many different fields to talk about, well, all sorts of things to do with intelligence. Because our goal at Cross Labs is to understand the mechanisms of intelligence, but also to understand about how does biology evolve intelligence? How does biology tell us about what consciousness is and all sorts of living systems? What about the mechanisms of living systems? But of course, these questions are so big that it's going to take many fields in order to solve these questions. So that's why we have a bunch of different speakers come and talk about their perspectives with all sorts of subtopics related to intelligence, biology, evolution, and then of course machine learning, because artificial intelligence is a really big part of the conversation right now. So Crossroads is also sponsored by Cross Compass, which is a leading AI research company here in Japan. So that's a lot of cross, right? But like we said, Cross Labs, Crossroads, Cross Compass, all of it has to do with the across interactions between different fields and industry and academia. So today is going to be in two parts, as usual. We're going to have a speaker, and today with us is Dr. Sam Gelman, and he's going to give a very exciting lecture about his new machine learning method called METAL. Especially what it's going to be used for is it's going to be used for, and it's currently being used for, predicting protein function based on not just the amino acid sequence, but all sorts of other information about these proteins. He'll tell you all about it. So that'll be the first part. And then the second part is, of course, a Q&A. So if you have any questions at all throughout Dr. Gelman's talk, please go ahead and add your questions either in the YouTube chat or in the Slido link. We're going to share the Slido link very soon as well. All right, so without further ado, I'd love to introduce our speaker today, Dr. Sam Gelman. He recently earned a PhD in computer science at the UW-Madison, that's the University of Wisconsin-Madison, and he is currently working at the Morgridge Institute for Research with Dr. Anthony Gitter, who is his PhD advisor along with Philip Romero. He also had two distinguished fellowships during his PhD program, including a predoctoral fellowship from the Pharma Foundation and a short-term traineeship from UW's Genomic Sciences Training Program. All right, so hi Sam, it's so great to have you with us today.
Hey Alyssa, it's awesome to be here. Thank you for having me.
Of course, I hear the weather is absolutely amazing in Madison right now, but it's a bit of a heat wave.
It's a little hot, but I'm looking forward to the rest of the summer still.
Nice, nice, nice. That's really cool. And also, how has been working in, I guess, like protein engineering? Like, what's that like on a day-to-day basis?
You know, it's a really exciting time to be doing research in protein engineering. Like, the field is moving very quickly, and there's a lot of interest from academic labs and from the industry, like startups and even big companies. So it's a fast-moving field. There's constantly new work being published. And keeping up with all the new work is like a job in and of itself, besides doing research. So yeah, it's a very exciting time, and I'm happy to be working in the field and happy to be here to share it all with you.
Awesome. That sounds good. That sounds really exciting and super cool. Well, I don't want to take up any more time away from your amazing talk. I can't wait to hear all about your amazing work. So without further ado, I'm now going to hand the stage to you. So take it away, Sam.
Cool. Thank you so much for the introduction. And hi, everyone. I'm really happy to be here. And today I'm going to be talking about biophysics-based protein language models for protein engineering. So proteins are a type of biological molecule which perform many important and diverse functions for living organisms. So for example, transport proteins like hemoglobin carry oxygen throughout our bodies. Structural proteins like collagen support our skin and bones. and hormone proteins like insulin regulate crucial physiological processes, and the list goes on. Proteins play a vital role in basically every process in our bodies, but proteins also have potential beyond their natural biological roles. And modern science has unlocked the ability for us to engineer proteins to fulfill new functions. Right. So, for example, in the industry, companies are developing enzymes that can efficiently digest and recycle plastics, which is really important for the future of climate change and the environment. In food science, there are efforts to engineer protein-based sugar substitutes that mimic the taste of sugar without triggering a glycemic response from the body. And in medicine, antibody proteins are being engineered to create therapies that target cancer and other diseases like COVID. And these examples just highlight a few of the real world applications where protein engineering can and is having a transformative impact. So every protein is defined by a sequence of amino acids. Amino acids are these smaller molecules that chain together to form a larger protein, and there are 20 unique amino acids in total. And a typical protein might be made up of something like 50 to 500 amino acids. So each amino acid has its own physical and chemical properties, and the amino acids interact with each other based on these properties, causing this amino acid chain to spontaneously fold into a distinct three-dimensional structure. And then this 3D structure determines the function of the protein. So to draw an analogy to natural language, amino acids are like words that come together to form meaningful sentences. And on the left here, I've listed some amino acids and their corresponding single letter abbreviations. And you might recognize some of the names like glutamine and tryptophan, which is often blamed for making you sleepy. And on the right is a figure showing amino acid embeddings from a model I trained, which I'll discuss in more detail later, but these embeddings are projected onto a 2D space and they're annotated with physical properties. So what this figure is showing is that the embedding clusters these amino acids together based on their physical properties. I'm going to use green fluorescent protein, also known as GFP, as an example throughout the rest of this talk. And this protein was originally found in jellyfish, and its main function is to emit green light in a process known as fluorescence. And GFP and engineered versions of it are often used as marker proteins in scientific research. And like all proteins, GFP has an amino acid sequence that folds into a distinct 3D structure. And the structure determines its function, in this case, brightness. So the version of this protein found in nature, which is known as the wild type version, has a brightness of 3.7. But by replacing one of the amino acids with a different one, we can alter the protein structure and consequently its function. So for example, if we replace the E in position 4 with an R, we create a variant known as E4R, which has lower brightness than the wild type. And we can also create other variants, perhaps with multiple amino acid substitutions that have higher brightness. So today we're gonna focus on a specific type of protein engineering that's very similar to this example. So we're given a wild type protein sequence and we wanna modify it to optimize a particular quantitative molecular property. And in this case, we're looking at brightness of green fluorescent protein, but there are many other proteins out there and many other properties that we may be interested in optimizing. So we might want to optimize the thermostability of an enzyme or the binding affinity of one protein to a different protein or molecule. Now, one of the core challenges in protein engineering is just the sheer number of possible protein sequences that can exist. So green fluorescent protein is 237 amino acids long. So just for proteins of this length, discounting, the fact that longer proteins can and do exist, there are approximately 2 times 10 to the 308 possible sequences. And this number is hard to comprehend, but suffice to say, even with the most advanced high throughput screening techniques in the lab, we would barely be scratching the surface if we were to synthesize and test these variants in the lab. So I like to imagine the engineering process as traversing a three-dimensional landscape with peaks and valleys. Peaks represent proteins with high function, and the valleys represent proteins with low function. And we can move through this space by modifying the amino acid sequence. So let's say we start with some wild type sequence here. We can introduce amino acid substitutions to move through the space until we find a variant at a peak that has our desired functional property. And I already discussed how this landscape is vast because of the number of possible sequences, but the landscape can also be complicated. There can be large swaths of sequences that are non-functional, and seemingly minor amino acid substitutions can sometimes have a large effect on function, which makes it hard to predict what amino acid substitutions will do what. So this is really where artificial intelligence and machine learning can be helpful. If we can train a neural network to accurately predict the functional effects of mutations, then this model can help guide our search through the sequence space and help us find variants with high function more effectively. Protein language models are a type of AI foundation model that are designed to learn and understand the language of amino acid sequences and how those sequences relate to protein structure and function. So the concept is really similar to natural language models that can understand the semantic meaning of words and sentences. Natural language models are trained on these large data sets of meaningful text that has been generated by humans over, decades, hundreds of years. And so this text has an underlying signal that the models can learn from about, what is meaningful in natural language. But what do protein language models learn from? So protein language models are based on the idea that evolution and natural selection throughout the course of millions of years have created a set of functional protein sequences, and that is the set of sequences that currently exists in nature, that has existed in nature. And by virtue of the process of natural selection, this data, this set of sequences, contains a signal about what it means for a protein to be functional. And this information encodes protein structure, it encodes biological function and evolutionary pressure within that collection of sequences. So protein language models are trained on databases of millions of these existing sequences that have been discovered in nature. And the training process uses techniques borrowed from natural language processing. So that includes autoregressive next token prediction and mask token prediction, where we kind of mask out certain words, and then we have the model learn to predict what word should go there. And that makes the model learn the underlying statistical distributions of the text. The same thing is done with protein language models, where we mask out amino acids, and then the models have to predict what amino acid should go there. So once a protein language model is trained, it can be applied to downstream tasks. So we can combine it with experimental data from the lab, for example, and use it to predict the functional activity of protein variants. The idea is kind of that, like, if our experimental data is limited, the PLM provides a useful inductive bias or a foundation to make the most of the experimental data that we do have. Now, I've made several comparisons to natural language, but there are important differences between proteins and natural language that should inform our modeling approach. So proteins are 3D molecules that exist in the real world. And unlike natural language, proteins are governed by physical constraints and the laws of physics. So these physical constraints have been studied for decades, and they inform core aspects of protein function, like how do proteins fold into 3D structures and how do they interact with other molecules. Uh, but existing protein language models don't use this information. And that really is the motivation for our study and what led us to create this method. Our idea is to bring in that information from biophysics and incorporate it into a protein language model by training a PLM on biophysical simulations. So, so far, I've talked about two different kinds of data. We have experimental data that we get from the lab, and that measures a specific functional property. And we said that how it's impossible to just test all the sequences in the lab. And we also have evolutionary data that we get from large sequence databases. But we're basing our protein language model on a third type of data, and that is simulated synthetic data from biophysics molecular simulations. So our method is called mutational effect transfer learning, or METL for short. And I broke METL down into 3 steps. Step 1 is to run molecular simulations to generate biophysical pre-training data. We're using a program called Rosetta to simulate protein invariants, and it allows us to compute biophysical attributes for each protein variant that we feed in. Step 2 is to train a model to predict those Rosetta attributes. And this gives our models an opportunity to learn about how mutations and sequence positions relate to each other based on these biophysical attributes, and to learn an internal representation of protein sequences based on those biophysical attributes and the input data. So this is analogous to the pre-training phase of evolutionary PLMs, except here we're learning from underlying biophysical signals rather than those evolutionary signals. And then finally, step 3 is to fine-tune our pre-trained model with that experimental data that we get from the lab to predict the exact function we're interested in, which is, whatever we tested in the lab, which we're trying to optimize. So there's a lot that goes into this, and I'm going to take you through these steps in a bit more detail to explain how the method works. So let's see, we have Rosetta, right? So we're using Rosetta to generate our synthetic biophysics pre-training data. And Rosetta uses simulation and optimization to compute these physics-based and statistical attributes for protein structures. So the biophysical attributes capture things like electrostatic interactions, solvation energies, van der Waals interactions, and hydrogen bonding. And generally speaking, these attributes are closely related to protein stability, which is important for a wide range of functions that we might be interested in. We sample variants from protein space. And then we use Rosetta to compute 55 biophysical attributes for each variant. So we're essentially generating training data so that we can learn the biophysical attribute landscapes across protein space. And because we are simulating our own pre-training data, we can choose what protein sequences we want to include in that pre-training data. So we came up with two strategies called local and global. And with the local approach, we want to learn a targeted protein representation that works very well for our protein of interest. So in this case, we're going to sample sequences to simulate around the space of that protein of interest. But with the global approach, we want to learn a general protein representation that works for any given target protein. And in this case, we're going to sample diverse protein sequences across all of protein sequence space so we can learn a general model. So let me explain a little bit more about how this works, and hopefully it'll become clear. So let's say we have a target protein in mind. So we're doing this local simulated approach. And so let's say we are interested in green fluorescent protein. Maybe we already tested some variants in the lab, and now we want to run simulations to generate additional simulated data to augment the experimental data that we have. So to run Rosetta, we need a protein structure. And conveniently, there's an existing one for green fluorescent protein in Protein Data Bank, which is just like an online database. But even if there is no existing structure, we've used AlphaFold structures with success, and so that should be fine too. So once we have our target structure and its sequence, of course, we generate variants of that sequence by randomly introducing amino acid substitutions around that sequence. So for local models, we generate 20 million random variants with up to 5 amino acid substitutions. And then we run those variants along with the wild type structure through Rosetta. Rosetta is going to simulate the structures of those variants and compute those 55 biophysical attributes for each of those variants. So at the end of this process, we have our local pre-training data, which contains protein variants of our target protein and the associated scores from Rosetta. The global approach differs in what protein variants we include in the training data. So like I said, with the local approach, we just generated variants of one protein, but with the global approach, we are selecting 150 diverse protein structures across protein space. And then we're generating 200,000 variants per structure to create a total data set of around 30 million protein variants. And the rest of the process is the same as the local approach. End of it, we're left with our global pre-training data. And again, the main difference is that it just covers a different part of protein sequence space. Okay, so once we generated our pre-training data, we train a neural network to predict those biophysical scores. And we're using a transformer encoder network that's similar to the original transformer introduced in Attention is All You Need. So at its core, it's really the same architecture that's used for natural language models, including LLMs like ChatGPT. Of course, those models are now using probably more advanced variants, but the core technology is kind of the same. So we feed in amino acid sequences as input, and the network outputs predictions for those biophysical attributes. And all these internal layers of the network are learning features throughout the training process that are useful for predicting those biophysical attributes. And then those features can be applied to downstream tasks like predicting protein function for protein engineering. So finally, the last step in our process is to combine our pre-trained model with experimental data from the lab. And to do this, we use a process called fine-tuning. So here I'm just showing the pre-training process where the model is learning to predict Rosetta energy terms or biophysical attributes. So we essentially make a copy of that network. We remove the top layer and add a new top layer. The reason is we do this is because that the top layer previously was very specific to predicting those biophysical attributes, but now we have a new top layer that's going to be specific to predicting the fitness score from our experimental data. We fine-tune the network on the experimental data from the lab, which just means we kind of train the network slowly on this new data to make sure it hasn't forgotten what it learned before, but also learns some new things from the experimental data. So finally, at the end of this, we're left with a model that is rooted in biophysics, but it can predict experimental function, and it can be used as a guide to direct our search for high functioning sequences in protein space. So in our paper, we did a pretty comprehensive evaluation of metal and other baselines. So I'm just going to cover some of those results today. But before we jump in, here's a kind of a quick summary of baselines you're going to see on the following slides. So of course, we have metal local and metal global, which uses an underlying biophysical signal that's trained on Rosetta simulations. We also tested ESM, or evolutionary scale modeling, which is kind of the go-to evolutionary based protein language model. So it trains on all of the diverse existing natural sequences. So you could say it kind of covers the global sequence space, similar to metal global. We also tested another evolutionary based method called EVE. EVE trains on local evolutionary data near the protein of interest. So in that way, it's more similar to metal local. So hopefully all of this makes sense so far. We have our method and we have these other evolutionary based protein language models that we're going to be comparing our method to. One of the main objectives of this research was to improve the model performance when we're dealing with small data set sizes, which happens pretty often in real protein engineering scenarios. So to evaluate these methods, we chose data sets that had a lot of data available, and we looked at performance as a function of training set size. So I'm showing the results here for an average across 8 data sets that we tested in the paper, as well as the dataset specific results for GFP, green fluorescent protein. And we measured performance by the Spearman correlation between true and predicted scores. So the higher the Spearman correlation, the better the model performs. And so most of the differences we see between the models are kind of at the small training set sizes. And overall, metal local and linear Eve, which the linear part is just because we used Eve in a linear regression framework, but you can kind of ignore that for now. But overall, metal local and linear Eve were the best performing models. Oh, by the way, another baseline that I didn't mention on the previous slide is just the linear. Linear is like a linear regression with a one-hot encoding. So it's kind of like the baseline, most simple model we could come up with. So what's interesting is that the two models that perform well, I guess better than the other models, metal local and linear Eve, are family specific models. They're local models that are targeted to the protein of interest. whereas the global models, Metal Global and ESM2, perform worse. So another interesting observation is that as we increase the number of training examples, all the models start to converge to similar levels of performance. So when we're training these models with tens of thousands of experimental data points, the pre-training we did is no longer as important as it is when we're training with less experimental data. Extrapolating beyond the train set. So on the previous slide, we looked at train and test sets that were sampled randomly. But we can also design more challenging train and test sets that kind of assess different aspects of how well the model can generalize when it's presented with biased training data. So what do I mean by biased? Well, for instance, some data sets might only contain examples with single mutations, but we may be interested in how well the model can generalize to multiple mutations. And in this case, the model would have to learn how mutations combine without seeing any multi-mutant examples in the training data. So hopefully that makes sense. We refer to this task as regime extrapolation, and it's actually the rightmost column in these plots. The REG is regime extrapolation. And Metal Local and Linear Eve perform better than the other models at predicting the function of multimutants when trained only on single mutants. At least for GFP, there's a noticeable difference. When we look at other data sets, the differences aren't as big. So we also tested other types of extrapolation. There's mutation, position, and score extrapolation. And I'll just run through them really quickly. So mutation extrapolation tests how well a model extrapolates to new mutations. Position extrapolation tests how well a model extrapolates to new sequence positions. And score extrapolation tests how well a model extrapolates from low scoring variants to high scoring variants. And score extrapolation is interesting because this scenario does come up in real world protein engineering, where sometimes there are very few examples of high scoring variants, and most of the variants we see are not very functional. And what's notable is that none of the models really perform that well on score extrapolation. So I think in general, this is one area where there's room for improvement. So a powerful aspect of pre-training on biophysical simulations is that the simulations can be designed to align with the protein function that we're interested in. So one of the experimental data sets we have is for a protein called GB1, which is an antibody binding protein. And the experimental data measures its binding affinity to IgG, which is a type of antibody. So to kind of better match the simulations that we're doing to this experimental function that we're interested in, we simulated the entire GB1 IgG structure complex, which is pictured here, rather than just the standalone GB1 structure. And we call our new binding specific model MetalBind. So performing this customized simulation allowed the model to learn a better representation during pre-training, which better aligns with the function of interest downstream. And as a result, we see a corresponding bump in performance compared to the standard Metal Local model. So I guess the takeaway from this experiment would be that that in the future, when simulations, as simulations continue improving, those can be brought into the machine learning framework and still benefit metal and other machine learning methods. So another aspect of metal we explored is how much simulated pre-training data do we actually need, and what's the relationship between experimental data and simulated data within the metal framework. So we ran this experiment where we trained models with varying amounts of simulated and experimental data. And we measured the performance for all the combinations of simulated and experimental data. So the numbers on this plot are showing the resulting Spearman correlation values, and the colored bands show where models perform roughly similarly. So for example, we trained a model with 1,000 simulated data points and 320 experimental data points, and we achieved a Spearman correlation of 0.67. We also trained a model with 8,000 simulated data points and 80 experimental data points, and it achieved a Spearman correlation of 0.66. So these two models perform very similarly, and if we do the math, in this case, adding 29 simulated data points gave the same performance boost as one experimental data point. And this is important because it's much, much harder to get experimental data from the lab than it is to simulate an additional 30 data points on a computer. So of course, this calculation only applies to this specific instance and depends on a number of other factors. Another result from this study is that there are diminishing returns for adding simulated data, and the benefits kind of taper off after around 128,000 simulated examples. So in the end, you might not need that many simulations to maximize your downstream performance. So finally, we put metal to the test to engineer green fluorescent protein variants and then actually test our designs in the lab. So we set up a very difficult design scenario where the model was only trained on 64 experimental data points, which is not that many. And those 64 examples had an average of 3.9 mutations, whereas like our designs had either 5 or 10 mutations. So the model was forced to extrapolate to higher numbers of mutations. We also had two types of designs, observed and unobserved. So where the models could only suggest mutations that were either observed in the training set or were not observed in the training set. And if the mutations were not observed, it's a harder situation because the model has to perform mutation extrapolation. And this plot on the right shows the resulting brightness measurements when the variants were synthesized and tested in the lab. So BT on the left stands for the best training set variant, and WT stands for wild type. And those are the two baselines we have here. And most of our designs exhibited fluorescence, and those are marked in green. Although none of them were brighter than the wild type, there were some that were similar to or brighter than the best training set variant that we had. So overall, considering how difficult this scenario was, this shows that metal can suggest functional mutations even when trained on very few experimental examples. And we're also currently running some follow-up experiments to test additional baselines and put these results more into context. So I'm really looking forward to getting those results back from the lab. So that kind of wraps up all of the main results for today. To summarize, I gave an introduction to proteins and protein engineering. We talked about protein language models and how they use evolutionary information to learn about protein function. Then I presented a new kind of protein language model trained on synthetic data from molecular simulations. And this is in contrast to existing PLMs, which train on natural sequences. And I would love to open it up for discussion. There's lots we can talk about. I listed some ideas here, but before we do that, I just want to acknowledge um, everyone who contributed to this project from the Gitter and the Romero lab. And I hope this talk was enjoyable for everyone in the audience, no matter your background or level of experience with proteins and machine learning. And if you're interested to read more, you can check out our preprint and our code, which is also posted on GitHub. And you can, of course, feel free to reach out to me with any questions after today's talk. So thank you very much for listening.
Excellent. Thank you so much, Sam. That was fantastic. It was really cool to learn all about the amazing work that you all did with not just designing metal, but testing it and fine tuning it and exploring all the different things that you can do with it.
It was a multi-year project, and doing all of those simulations and experiments, it takes time.
Especially that one figure that you have, that's that grid where it has the experimental like the experimental data set size and then the simulated data set size. I honestly cannot imagine trying to run all of those simulations. Like, that's a lot of compute.
Yeah, absolutely. And you know what's funny is behind the scenes, I didn't even mention this, but like we test multiple replicates to account for randomness. So like one of those squares probably has like hundreds of models trained behind it to get that result. So yeah, I really should also acknowledge the Center for High Throughput Computing at UW-Madison, because I couldn't have done it without their help and their compute resources as well.
That totally makes sense. And so this was all at UW's servers, right?
Yeah, that's right. This was all running on UW resources. And at Morgridge, we actually work very closely with University of Wisconsin-Madison, as you know. So we still are running with running experiments on those servers. But
Nice. Oh, nice. Yeah. Well, what experiments are you running now? Just curious. Say.
we're doing we're doing paper revisions. So we are testing some new baseline methods and just getting some additional results. But yeah, we also started a new project kind of based on metal, but we're in the very early stages. But the idea for that one is also really cool. But essentially, we're I said there's 20 unique amino acids, and that's true. There's 20 to 22 that are found in nature, but you can synthesize hundreds of amino acids chemically in a lab. So there can be hundreds of amino acids, which totally changes the language of proteins. And so far, there has been like very little work in that area. So we're kind of interested in taking metal and applying it for these so-called non-canonical amino acids.
Hmm. Oh, that's very interesting. Curious about what would be the uses for these non-canonical amino acids. Like, what are like the industrial uses for these sorts of proteins? Or, I don't know. Like, I'm just super curious. Like, what would be the motivation behind that?
Yeah, so non-canonicals, they're used in therapeutics. So peptide therapeutics, which are which are chains of amino acids, but they're not quite long enough to be considered a protein. And they're not quite short enough to be considered a small molecule. Um, there's a lot of potential there to integrate non-canonical amino acids because they have different physical and chemical properties, and can potentially help develop better therapeutics.
Gotcha. Oh yeah, that would be really interesting to yeah, super interesting to explore. So I think I'd be really curious to see how those results pan out.
Yeah.
Nice.
Um, yeah.
um I so at this point, I think that I'm going to pop on over to the Slido because we had some questions for the audience, and let's see what they have to say. All right. So the first question talks about LLMs and hallucination. So, as language is within our dimension of understanding, I can tell that an LLM hallucinated. So, how can you evaluate the outputs of this model? So, I guess in a sense is like we it seems obvious when an LLM hallucinates, but maybe it's less obvious when another kind of machine learning method like metal hallucinates. How can you tell when or if it does?
Um, that's it's a great question because in natural language models, I mean, we can like the question says, I mean, we can Google and check the actual facts and see if the LLM is hallucinating or not. But even with that, it can still be hard to evaluate LLMs sometimes for whether they're hallucinating or not. But with proteins, it's even harder because with generative models for proteins, you really don't know whether the output you're getting is kind of a valid protein that will actually function unless you go and synthesize it and test it in the lab, which can be a very expensive and time consuming process. So I know that like just the question of how do we evaluate these generative protein models is kind of a big one right now in the industry. And there's not really a good answer for how to do that yet.
Mm, that seems tricky. Do you think things like Rosetta, or I mean, sorry, the biophysics simulations could help with it? I'm not really sure. Just curious.
Yeah, I mean, you can feed a protein variant into Rosetta, and Rosetta will use it, biophysical knowledge to kind of figure out whether maybe this protein will fold into a 3D structure, whether it'll be stable. And yeah, that can definitely be helpful. But as far as like, we want our protein to do X, Y, and Z, like bind to another molecule, or we want it to, you know, be useful as a therapeutic for disease. That is a little more complicated and really can't be done to a high degree of accuracy with Rosetta. So like ultimately we can kind of try to observe, the sequence that is generated and learn more about it through these computational tools. But the ultimate test is still, we got to synthesize it, we got to test it, and that just makes it challenging.
Mm, that totally makes sense. And speaking of synthesizing and testing, I'm just curious, like, how long does it take to calculate? I guess I guess like how long does it take to generate this experimental data in the lab? Because you talked about the experimental data sets for training, and I like, is it expensive? Does it take a long time? Can you tell us a little bit more about that?
Yeah. So I guess the answer is, in short, it depends. But we work really closely with our biochemistry collaborators who are amazing and do all of these wet lab experiments. But it, but the scale to which we can generate data in the lab is like orders of magnitude slower than simulating on a computer, at least the way we did it with Rosetta. Um, so on the wet lab side, there has been a lot of developments in high throughput screening technologies. Probably in the last like 10 years or so, there's been like a big surge of what's called deep mutational scanning techniques, where scientists can test, up to 1 million variants in the lab in a single experiment. Now, there are definitely limitations to what functions we can screen for and what proteins are suitable for those types of high throughput experiments. But certainly that does generate a lot of experimental data. But there's also more complex phenotypes that we may be interested in that take just longer to determine. So like if we want to know whether a potential therapeutic protein or drug, has side effects, or just, and we want to test that in a mouse model, for example, well, that takes time. I think it could take weeks just to test a single variant in a mouse. So like it's a big scale of like of potentially like how long it takes to generate wet lab data.
Mm, yeah, that makes sense. And you said that there's also some limits on the kinds of functions that you can test for. Like, what are some of those limits? Just curious.
Yeah. Well, there's actually a lot of functions you can test for. You know, data sets that we have looked at things like binding to other proteins and stability. I see those types of data sets pretty regularly, but I probably don't have the in-depth knowledge of like the wet lab to like answer that fully. But if it really depends like what phenotype you're interested in
Mm.
and whether you
So
can design an assay to test it.
right. Yeah. Oh, that makes sense. That's a lot of yeah, sounds really difficult. So thank goodness we have like these computational methods that can sort of fill in the gaps where needed. Um, so I'll go to the next Slido, which talks about like of course the computational I guess like prowess of this project these projects. But so the number of possibilities of the different amino acid, like the variants for a single protein, is absolutely ridiculous. So, just out of curiosity, if we were to brute force all of the different combinations of the amino acid sequences, how long would it take with today's tech? Just for fun.
Um, yeah, I guess it would probably take longer than any of us will be around to see the results. So um, it's all a game of like picking, exploring this vast, seemingly it's not actually infinite, but because it is, we can quantify exactly how many variants there are, but this massive space, like exploring it intelligently and putting our resources into the paths that are going to give us the most payoff. And that's kind of what these machine learning methods and AI is trying to help us do, um, to kind of navigate that space. So hopefully that answers the question.
That totally makes sense. Yes, because it's not just about the brute force computing tech. It's also about the tech in terms of like AI guiding and providing insights on like how you navigate. Yeah, totally love that. That's great. And also to follow up with that, like how long did it take on the UW cluster to generate the local and training data?
Yeah. So I have the number somewhere, but I don't know them off the top of my head. But I'm pretty sure I measured it in compute days, but it was quite a large number. But the thing is, with the resources that we had, we distributed the computation in parallel across like 9,000 CPUs. Um, and when you do that, like in real lifetime, it ends up taking like a week or two to generate like 20 million local training variants. Um, but yeah, I'm gonna have to caveat that with, again, it depends on like what proteins we're looking at, how long it takes. That's one of the experiments we're doing right now, to follow up on our preprint, is to kind of quantify exactly um, the computational cost of running those simulations and training the models, and to see like how accessible it is for, you know, any scientist to take off the shelf and run on their own hardware.
Mm, right, right. That's true. And over 9,000 CPUs. That's absolutely insane.
Yeah, it's really cool. It's so that was also through Open Science Grid, which is this consortium of public institutions that are donating CPU time that, you know, not every CPU is active 100% of the time. So they give access to other researchers, and we were able to take advantage of that through Open Science Grid.
Oh, nice, nice. No, I'm curious, like if you're a hobbyist or you're a researcher at a institution that doesn't have large computational power, are they able to access the Open Science Grid? Do you know?
I don't know.
Mm.
I know if you're at an institution like a research university, you can, but there may need to be a US based connection. So there's definitely limitations. I just don't know what they are.
I see, I see. So everybody who is so if everybody who's watching and is interested in getting extra compute time, we encourage you to look at the Open Science Grid website to see like what the requirements are, because that's over 9,000 CPUs. That's amazing. That's a dream for sure.
Yeah.
Great. So I guess like one final question before we wrap up. What like for you personally, what is your hopes and dreams for how this machine learning method metal gets used, and especially for like industrial uses? Because we had talked about a few of them before, but is there one like personally that really sticks out to you and really gives you inspiration and hope?
So I said at the beginning of this talk that like it's a really exciting time to be doing research in this field, and part of that for me personally is just because of the where we are in the potential for having a real impact. So we're right at the cusp, or I mean, it's even happening right now, where these methods are being used in the industry. Um, so that gets me really excited. And I mean, there are so many different cool applications. One of them I didn't mention was um, actually, I did mention it, the sugar substitute. I thought that was really cool. But I think there is just, especially in the like therapeutic side, so much potential that's starting to be realized, and I don't know, that gets me really excited to continue working in the field.
Nice. Yeah, nice. It's very exciting as well, and I can't wait to see how the field develops and how metal gets used for all sorts of different applications. It's very exciting, and so glad that we get to hear your adventure and that you're a part of it and all that sort of stuff.
Thank you so much for having me, Alyssa.
Yeah, thank you so much for joining us today. This was absolutely fantastic. And thank you all so much for tuning in to today's Crossroads. And I'll say it again, don't forget to like and subscribe so that you'll be first to know when the next Crossroads will be, and also all of our other content as well. So if you're watching in the future for recording, thank you so much for joining us. Please check out our other content, and don't forget to visit crosslabs.com. Crosslabs.org to see all of our cool research and projects and see all the cool projects and research of our collaborators as well. And yeah, thank you so much. We will see you next time. Bye.